I am Clarinda.
I'm Carolyn.
And this is the last time we're going to be saying this, but we are the GenSpace Creative Program Coordinators, and we, and this is our last week in that position, but we have started an artist in residency program in collaboration with the GenSpace staff and community, and we have had a cohort of 3 months, and they're coming to an end, and that's why you're here today.
Two wonderful humans
Yes.
that you will hear from and get to experience their stuff after you hear from them. But we are staying on to help run a second cohort of the Artists in Residence. So if any of you or anybody you know are interested in applying, go to the GenSpace website, look up Artists in Residence, and please apply, or encourage someone to apply. I don't think we should belabor this.
No.
We're excited to introduce the two artists. Io and Alvaro have been amazing, and we're going to let them give a little bit of their own introductions when they come up. Alvaro, you're first, right?
Yes.
Okay, so everybody please welcome Alvaro. And don't forget to project.
Yes.
Carolyn, you want to turn the lights off?
How should I do it? Well,
Alvaro. Alvaro,
Do you want the
can
lights
stand on that.
Do you want the lights
but
off?
for the
Oh, I'll do it.
Do you want some? Do we need lights for the live stream? Are we okay? Taylor,
Huh?
do we have lights for the live stream?
No, it looks good like that.
Okay, great.
well,
Don't fall asleep.
hello everyone and welcome. My name is Alvaro Azcárraga. I'm going to be presenting a little bit about my project called the Frankencorn Project, which is still a working title, so I'm not like completely married to that. So yeah. Oh, well. I am Alvaro. For those of you who weren't here last time, I'm going to go over a couple of the slides and just talking a little bit about my practice. This is my name. I am a queer bioartist born in Mexico City and based in Brooklyn. I come from a molecular and cell biology background, graduated from UC Berkeley. And my work examines the post-natural, specifically looking at the artifice that is embedded within the term natural. I've been working with maize, Z-maize, aka corn, for the last 7 years. And by growing maize in botanical gardens and greenhouses. And I also work alongside plants with a focus on the history of scientific colonialism, dealing with issues on invasive species inheritance. This is a little, I just wanted to give a couple of slides on like past work. This is the Huitlacoche project based in Buffalo, and I did this whole like 12-course dinner where I prepared like each course was like related to each of the steps in the domestication process from teosinte to maize. I'm going to be going through all of this in more depth. So the Frankencorn working title still a little Frankenstein reference. But yeah, Frankenstein and genetic engineering, there's like this kind of like history as well as like this really interesting book, an article on how like scientists and like Frankenstein, the narrative of Frankenstein, like Mary Shelley's novel, as well as movies in the past, have been kind of like tapping into the sphere of genetic engineering as well as genetic purity that exists within like our within our research, and how like putting trust on specific scientists to do like this research is either good or bad. And there's this really good book called Legacy of Frankenstein. The monster is the one in the lab coat, and you can check it out. So yeah, just looking around Frankencorn on Google, this is kind of like what comes up. This anxiety alongside like genetic modification in plants and how they embed into like the human and like the human collective narrative. I'm also going to talk a little bit about grafting because that's part of my project. For those of you who don't know, grafting is when you attach two different plants together from two different species and then they like slowly merge into one. If you're not familiar with the Tree of 40 Fruits by Sam Van Eycken, you should definitely check his work out. He does an amazing project where he like puts 40 different fruit trees on one specific stem, and they are growing all over the US and some internationally. Just highly recommend his work. So I was also inspired by this Biombo de la Conquista de Mexico, which means it's like a room divider of the Mexico conquest. I am from Mexico, so I was like thinking a lot about how these functional objects and like lineages get told through like functional objects. So this is one from the 17th century that depicts the conquest of Mexico in like both sides, as well as like a little map of what Mexico used to look like. So these were, a couple of these were created during the 17th century to kind of like depict this lost history that was like whitewashed by Spanish conquistadores. And this was just kind of like a different depiction of Mexico's history. So just a quick maize history. Corn comes from the Proto-Germanic corn with a K, meaning grain and mature. So this kind of like lost all sense of like where the word comes from. Maize comes from the Taino word maíz. And tlaoyi is actually the Mexica word for maize, which is like what the Mexicas, like what Aztecs are in current day Mexico. Teosinte was domesticated from like 6,000 to 10,000 years ago. Closer to 9,000 is like the current scientific standpoint. In the Balsas River Valley, in current day like Puebla, Oaxaca, if you're familiar with that area. And these are just like some fossil images as well as some. So it was like domesticated from Teosinte, which you can see over here. And maize is like on the right. So if you can like cross them each other, and this is like a maize Teosinte hybrid. And yeah, so this is just kind of like a brief overview. Just a map of Mexico. The Balsas River Valley is over here. The United States is over there. And this is just a little bit more into the Teosinte research, which I won't bore you too much with the details. So just as another point, maize grows everywhere in the world. Honestly, you can see from this map, which just shows like food calories supplied from maize. That is, yeah, just anywhere there's like fertile land, maize grows. It was actually one of the first plants that was like shot up in space to do like some research experiments, and it grows anywhere, even Antarctica, for research purposes, obviously. I was also really fascinated by the TIN1 gene, which is a tillering gene. So basically, maize, as we know, like grows in one single stalk. Teosinte grows in a branched variety, and I was kind of like looking at this gene. I was like looking at these like branching aspects of maize that got lost through domestication, and I was just able like to play kind of like looking at lineages, looking at maps, as well as just kind of like how these biombos, is like these room dividers, kind of like work, and just kind of like looking at these like functional elements to kind of play around through this residency. And this is another, it's just phylogenetic tree of maize, Teosinte. I won't go into too much detail about this, but just I was looking at all this kind of like information and research. I can talk more about this if you're interested later on. Just showcasing the variety of Mexican maize that exists. Actually, I saw this printed in Dave's lab over here. I just wanted to meet, so now we're gonna go a little bit more into the research. We're gonna meet the seeds. So Teosinte, as I have talked about it before, it's the ancestor of maize. Then I'm also going to talk about the Mexican land races, which are like varieties that have been cultivated by farmers for generations and have like a genetic identity, a distinct genetic identity. And then B73 was released in 1972 by Wilbur Russell, and its descendants are all like grown all over the world. So it's this kind of like a hyper-domesticated American maize that is like now used as the essential maize for genetic research all over. So now, what is plant callus? Plant callus is a growing mass of unorganized plant stem cells made through plant hormones, mostly auxins and cytokinins. And so basically, plant callus is kind of this plant stem cell that kind of like produces these like clumps of cells that either produce shoots or roots. And I was kind of playing around with this idea of this research that I found and kind of like looking at how this can portray like different areas along with the research that I was doing previously, the historical context. So the idea for this research, which obviously was very ambitious at the time, but this was the original goal, was to kind of like take a little piece of the Teosinte callus, take a little piece of the Mexican landrace callus, take a little piece of the B73 callus, create one single callus, and then differentiate it to like a single plant. So ideally, this plant would have all three varieties in one single organism. And obviously, like making reference to the Chimera, making reference to the Frankenstein, making reference to a bunch of research that I actually have a couple of books that I just wanted to show that were very important in this part, which I just highly recommend the Graft Hybrid. It just came out recently, and it speaks about grafting as a practice historically, as well as genetic research. It's for those of you interested in this, it's an exciting read. A lot of like scientific drama involved. And then Plants from Test Tubes is one of the micropropagation like handbooks that a lot of like plant researchers, like plant hobbyists from home use, and it's just a really good read. And it has like it's like very dense like numbers based. So I just wanted to talk a little bit about my callus protocol, where it was just, and you'll be able to see all of this later on in the second space, and we'll just be able to like put on gloves and you'll be able to like experience all of this hands-on. But basically, this is kind of the protocol where it was like a medium that was developed, which basically was agar, the hormones, and some nutrients that the plant needs to grow, and then that like kind of induced this plant callus formation. And so I just wanted to like highlight the community plant cell culture, which is these two like YouTube sources that were like really helpful. I highly recommend their videos. They're really interesting people. Plants in Jars is like a home-based kind of a YouTuber that is based in Florida, and I actually like wrote thanks to her today, and she responded, which I was like very grateful for. And Plant Cell Technology is a Mexican enthusiast that does a lot of like, plant cell culture, and he actually sells a lot of these like, materials for like home-based plant cell culture. And now a little bit about like images on like how this was done. The 3 images on the right were taken at Dave's lab, who's sitting right over there. And as you can see, it's like this kind of gel in a petri dish that grows a clump of cells, and this is the callus over here. And then it starts to differentiate into like different leaves. So it eventually becomes this like mass of cells and leaves, and it just found that like super interesting aesthetically. But it's also very commonly used for genetic engineering, where it's like basically this callus is shot with a gene gun, that's literally what it's called, where small fragments of DNA are like shot into the cell, and then that's how like transformations occur, or through like bacterial through like infection. That's another way. These are just a couple of examples that I made, so that you'll be able to see them over there. Some of them survived, some of them didn't. As people who work in this lab know, there's a lot of things that get infected, and you'll see a couple of examples of that later on. But yeah, this is B73 that's growing in the gel, as well as a couple of like leaf samples that were like growing to create callus, and the Sur Bonita, which is another variety of maize of the Mexican landrace that was like actually grown and is still surviving, so you'll get to see that in person. And just a couple of other containers and like seeds that were sprouting. Yes, a lot of contamination occurred. It was a constant kind of like battle. Of course, using the community lab, this is kind of what we have to encounter. And even while you're being like as safe as you can, it's just a matter of fact of like the practice. I still was able to get callus, which was the original goal, so I was like very happy. This was confirmed by Dave, which was very grateful that I was able to like get like at least one of them. This was B73, so it was like one of the varieties that was like easiest to get callus. And then I was able to like look at them through on the microscope, and you can kind of like see how they're like little clumps of unorganized cells, as the definition kind of entails. Which then, as time goes on, it transforms into this like green-like structure, and this just takes a little bit more time and care. But my callus got infected, which then meant we lost a lot of stuff. So yeah, you can just kind of like see the process of extraction and as well as of plating. It just takes a lot of time, and even in the hood where we kind of try to keep like a sterile environment, we try the best to just kind of like keep a little. So this is just a little time lapse that I was like recording of me in the lab, trying to be as safe as possible while still getting up several times, which I shouldn't have, but it's fine. Yeah. And then another side experiment, which it was really nice to kind of have both the actual science, the science of the lab, as well as on top of the like kind of practical crafting at home. I was able to like develop this paper husk recipe, which took a lot of failed attempts until I found this kind of really good protocol that actually worked, where through like fresh leaves. So basically the maize that you ate today, the leaves were used for paper making. And basically through just kind of like a regular paper making process, you kind of like have to like sift the fibers and process the fibers so that they make a flattened, stable paper. And it's actually pretty sturdy and really interesting to see how it like develops. And I actually was able to include a couple of slides at Cold Spring Harbor with Dave Jackson, who's over here as well. Dave was like nice enough to lend a couple of rows over at Cold Spring Harbor. And I'm actually planting a couple of varieties, and these are just some images from Dave showing that there was a positive like, sprouting in the area. So at least they didn't, they survived. So these are all like different experiments that I'm doing row by row, where each one has like a different variety, as well as one that's like open pollination, which I'm calling, which is just going to be kept open so that it like cross pollinates. They all have different like sprouting rates, but I'm just trying to like keep it a little bit. And the goal would be to kind of cross the Tocine Gamkite and maize, and that's kind of like the goal that where I'm like heading. Well, I just wanted to thank everyone for that I will Dave, Trading Biotech Friends, and Casey and the whole Space team for these last couple of months. I might have rushed a little bit through the slides. Sorry about like speaking so quickly, and hopefully you were able to understand most of what I say. But I'm happy to answer any questions later on. I think we're going to have questions after
Yes,
my turn.
well, we can do 5 minutes of questions right now,
Okay.
since we have 5 more minutes of your time.
Perfect.
Does anybody have questions for Alvaro?
Thank you for your presentation. Super interesting work that you're doing, especially the planting that you're doing at Cold Spring Harbor Lab. Do you think that that will produce corn that is a chimera? Like, will be like on one, can you explain that a little bit
Yes.
more? Will like one plant producing different types of corn, or will it be like one corn on the cob at a different?
I have this one slide that I skipped, but basically how corn fertilizes it has the tassels that you see on the top. That's at the very top of the plant, and then it has like the little hairs on top of the corn that you see when you like are pruning it off. Basically, the tassels produce pollen, and then the pollen goes to the little hairs, and then that's how like each individual kernel is pollinized by each individual hair. So you can kind of, we were talking with Dave, that you can kind of like comb them and kind of select them to see how you can kind of play around with how rows get like fertilized into different species. This would not be a chimera, it would just be like a cross between like different species, within different varieties of maize, not species, because it's the same species. So this would kind of be like a more specific like playing with the differentiation between like the domestication and breeding process. That's kind of it. Other questions?
Moving through the history that you're speaking about over that 10,000, 12,000 year period of domestication, has there been a gain and fall in nutrients as we made the species more easily for our agricultural processes?
I'm not super familiar with that aspect of a kind of like nutrient aspect of it. It's mostly kept because also there's, then you have to go into like the two types of maize, which is the white maize and the yellow maize. White is usually starch heavy and yellow is like protein heavy. So the yellow one is used for animal feed mostly, so it's kind of like the American version of like most of the grain that's grown in the United States either goes to animal feed or ethanol. But most of the grain grown in Mexico is mostly used for starch purposes. So that kind of division has like a different nutrient kind of density to it, but it would be difficult to kind of like talk about like starches and proteins where you're like talking about two almost completely different varieties.
Thank you. How are you envisioning the final presentation of the project?
Yes.
You're talking about the jumbo,
Yes. So once you go into the little room, you'll be able to see kind of like the components of like what I'm envisioning. So ideally, there's going to be like there's a little wooden structure that I'm going to be working with that is basically going to fit these like Chinese papers, which that's the goal. And then there's going to be like some sort of element that encompasses the actual hybrids themselves within the screens. So ideally, it's going to be this kind of gradient of domestication that you like walk through these like movable walls that then like talks about the original step of the Conquista de Mexico, like that I was talking about before. So the idea is to kind of like connect those two areas. Which thank you for that question. That was really good. Yeah.
Wonderful presentation.
are you planning on doing any grafting with your grown corn that you're growing in this garden?
So interestingly, maize, since it's a grass, so it's part of the grass family, it can't be grafted. It's like one of those like things that it's technically not possible. There is an instance that embryos were potentially grafted, but that's kind of like a kind of new kind of science where they cut different embryos and literally super glued them together, and that kind of created a little like two-sided graft. But like you can't cut maize that's grown and then like attach it to maize because it's like a grass, a monocotyl, which is like a different type of structure that like the plant doesn't allow for grafting. So the idea is for the cells to then be grafted, so the cells grow together. It might not expand into like the field, but it will be like conceptual kind of togetherness.
Okay, one more question.
Oh, one more question? Yeah.
How has your relationship with maize or corn changed by working with
That's a very good question. It's changed quite a lot because I am, as you can tell, a little obsessed with the topic. It's been like my passion project for the last couple of like 6-7 years, and it's just this kind of like area that the more I like dig into it, the more I find, and just like the history of it like keeps on like giving more and more and more. And it seems like it's an area of research that it's so commonplace for like so many of us, especially in the U.S. It like grows in the most of the country, but we don't even see it or we don't even acknowledge it. And when we do acknowledge it, it's like for Middle America and this kind of like aspect of the Corn Belt. But then this kind of Mexico Corn Belt kind of distinction has always been interesting. And I obviously like grew up in Mexico, but when it came to the U.S., that whole like transition of like how people, like the general public, like sees maize or corn, it's just like super interesting. Like even learning about like telling people that I'm like researching maize and then showing them a picture of corn and them not like connecting the dots that they're the same organism was like, oh, so there's something there. Like people are like not really like paying attention to the most widely grown plant in the world. So it's this like global superstar that like isn't visible in a way.
Thank you. Before y'all get a chance to like get personal with some of the seedlings that we've been talking about in the paper, we are now going to hear from Io Shaboy.
Can we get all ourselves, please? Hi, I'm Io, and this project, yes please, yeah. This project has been kind of this exploration of being in the lab for 3 months and really thinking about what is SCOBY and like making giant SCOBYs like this one and just really like deepening my relationship with something that I was calling an entity that is now becoming like an organism that I'm putting myself and maybe other parts of you all inside of. So the title for both theirs and mine comes from this quote from East Montu Gwendolyn, where she's talking about the case for a global ceasefire. And she says, "This is the way I pray for the sun and for Palestine, with this heart beating inside of me. That is both theirs and mine." So before starting this residency, when I was writing about how do I even apply to this space, I asked, what does it mean to think about biotechnology when we're in this time? So much war is happening, a lot of people are dying. How do we think about moving forward into the future, but also hold the space for people that are dying, for death that is occurring at the hands of white supremacy, capitalism, of settler state colonialism, and putting all of that into this is why this matters, because we're questioning what does it look like when structures are restructuring, when things are falling apart, and wondering like where do we fit inside of them. I don't have answers for that, and I'm sure a lot of us in this room don't have those answers either. But I've been wanting to chew those bits and continue to like put them into what this art is. That is an extension of myself and an extension of my ancestors. So this is my SCOBY heart. This has been this process of, before even getting to this point, just questioning what is the agency of these microbes that I'm working with? How do I separate my human notion of understanding from how I connect with them? Do they think on their own? Do they feel? Separating that from how humans think and feel. And what does it feel like to hold something that is beating in your own hand? And this project is based from, I put a lot of my family into the work. My aunt needs a heart. My grandma has 7 stents in her heart. So thinking about how can we think about the future of healthcare? How can organisms kind of stand in as a way to see the things that are inside of us that we can't necessarily like touch or examine? And what does illness look like? And how do we kind of bring that into a space that becomes intimate when you're holding something that resembles something that feels like human? So this is a vibration test. So my finger is hooked up to a heart sensor that's connected to an Arduino board. And this is my first time actually touching technology in a way where I'm connecting it to something. This is all thanks to my mentor, Pia Kelsey O'Neill. She's a neuroscientist at Columbia. She's ending her stint there. But without her, I couldn't do any of this. Because I was avoiding touching technology as much as possible, even though this work is very grounded in technology. Because I couldn't figure out the separation between where do I begin and where does this technology begin, and are there separations? Okay. And a lot of this process, also this is not a linear timeline of how this project has worked, but this is a torso of my body, I'm calling SCOBY body, where when I was kind of questioning what does it mean to enter into a digital space, to use technology in different ways, are parts of me being fragmented as I slip inside of virtuality. If I am breaking parts of myself off, how do I carry them with me? How do I carry that lineage perhaps on my back, but honoring the body and the bodies of the people that came before me? So kind of layering the SCOBY on this form has been this ritual of trying to connect. How does ritual connect to lab work? How does ritual connect to care? And how do I honor this vessel as I exist? And these are some intergenerational mothers, where Mother 3.0 is a mix of Mother OG and Mother 2.0. And I've been able to scale up in the lab and thinking about what does it mean when these microbes now have a new space to live and grow and to kind of coexist together. How does this lineage kind of mark the way I'm thinking of lineage as I honor my family through my art practice? These are just some getting familiar with the lab, touching SCOBY, growing them, and this is before the scaling up. So just thinking about how do I grow SCOBY in Petri dishes within the lab? And I kind of thought of this process as like transplants. Each time I created a Petri dish with SCOBY, it was transplanted from the mother before, and continuing the cycle of intergenerational weaving of all of these lab SCOBYs became that SCOBY heart. This more tests, initial setups, I spent a lot of time looking under microscopes because when I was in grad school, I always wanted to know what was inside of the SCOBY. Not just how do I grow it, how do I tend to it, how do I nurture it, but who are you? What are you? And this is just an example of me starting to grow the SCOBY and like use different equipment and just see how do bubbles affect the way SCOBY is growing? How does water, distilled or tap water affect? So that will all be recorded also on the website. I'm just like in a process of digesting, so that's not here yet. So these are some of the microscope tests that I spent a lot of time looking at. Thinking about when I use a microscope, there's a lot of waiting and a lot of wanting, where you don't see what you want quickly, unless you're looking at perhaps the callus and you're, oh, there it is. But the microbes themselves were kind of like waiting for me to focus the microscope and kind of showing themselves as I spent time with them. So these are distortions of the original images. This is kind of just like my eye looking down. What happens when bacteria starts to infect the SCOBY body? How does that bacteria start to layer and continue to like adapt to the form? These are some of the microbes. So over here, this is what the yeast looks like. some of these other small bundles are the bacteria. But this was the first moment where I saw, oh, this is my mother. These are some more tests. So I started to look at, oh, how are the different bundles of the microbes like growing in different ways? Does the water affect that? And just kind of keeping like a loose process that wasn't very like rigid with science or rigid with method, but still like organically finding my way and just like noting the shapes. And right here, this is a dye test where I first saw how the fibrils of the SCOBY start to come together. So some of these clumps are just masses of the bacteria, but right here is where I started to see, oh, this is how the SCOBY grows. When the fibrils start to come together, it started to make me think of, how do I connect to community? And I spent a lot of time talking to people in the lab about connection and community, and that was the extension of, where does my heart beat and connect and meet with others? And seeing these fibrils, we're like, how do we connect and meet with each other? This is the link slide. So now, that was a pause for a moment back. That was good. So this is where this project kind of originated. I was calling my first SCOBY Prometheus. We're going back in time now, so we'll see Prometheus. But this was the origin of the SCOBY heart, where I started to grow the SCOBY in a massive glass jar and would just use embroidery cloths to stitch it together. I didn't know why I was doing it, but I kept doing it. And something about it was like, just keep doing this. And now we know why. I made a heart of some sort. And then, so this is kind of hard to see, but this is where I was starting to weave ritual into, how does South Brooklyn move through me? How do I walk the waterways along Red Hook when I still lived in this area? And how do I honor the moon when I'm on the land? So this is me wearing a dress that I stitched SCOBY into the side of, and I walked on the waterways, and it says, nature reveals herself to science, blood moon ritual. The kind of putting my queer body in a space that is like touching this gown that my mother wore, touching the SCOBY that I grew, that I started to weave in to think about, how do we mend intergenerational trauma? Thinking of that made me think of, how do I let this light, that is this darkness, how do I find this light inside of me, where I'm consumed with darkness, and thinking about, how do I reconnect to ancestral lineage? How do I use the SCOBY as kind of like a guidepost or a lighthouse that I am moving toward? And this is the question, can generation be mended across, can trauma be generated, you can read, maybe, can trauma be mended across generations? So this is when I started to take time to just slow down and stitch and mend and to kind of fix the holes that started to form inside of the SCOBY, but to really question, if my body was once inside of this dress when I was a baby, how does my body now, this timeline, fit back into that dress? Knowing that there are new conditions placed on it, there are new things inside of me that are like changing the way I respond to those conditions, and how do I honor my mother, who wore this gown until it tore, until she birthed me, until I birthed this heart. Thinking about how to weave ritual into just this practice, this is where I started to question, what does it mean to be a mother? How am I my own mother? How do I take all these things that have been passed down and kind of digest them? How do I burn and release and continue to like honor this land? This was created in Red Hook as well. And I started to really connect to this artist named Ana Mendieta, where she's really honoring the land through the way she showed devotion and how she carved her body and other forms into it. I question, how can I carve my body with the forms or just the quotes that I have floating inside of me? This is something I call consuming mother, where a mother starts to consume the parts of my body that I have been breaking off, and we are still going back in time. So as I'm deconstructing the torso, I'm also taking my fingers off and wrapping the mother around it. This is the birth of Prometheus. This is the SCOBY that originated all of this process. This is a pause. And this is kind of questioning if I am breaking my body apart, how do I use the environment to connect to myself, to make models of. I took this moss from my backyard and put it into this mold that I made of my foot. This is a bioplastic made of alginate. And the little dish that I put it in kind of reminds me of a petri dish. This is where I'm asking other forms to hold my form. This jar is made with the used alginate forms that I used to shape my mouth, shape some of the little skin flaps to make this foot. Just kind of thinking, how can emotions be stored in other places, and how is this still trauma being passed along? I used these molds of my lips and stuffed some of the moss in the backyard in my house inside of them to some question, how does the earth move within me? And how do I start thinking of consumption and the parts that I am now connecting my diet to this art and the environment around me? And now this is the origin of where the body came from, where the cast was first made. The arms were broken off, the feet were broken off, but then the materials around it started to rebuild. My fingers, some mouth molds. This is where I started to think more about how do I fold bell pepper seeds into these lips, more moss, more rice. And that's why the food that I made tonight is a part of this process as well. How do we think about what we're consuming? How does that form a memory? How does that connect us to this space? And how does that show up in the art? And just this question is a cycle of regeneration like rebirth? This constant ongoing cycle of forming the SCOBY and reforming the SCOBY, of having organisms hold my shape, of having that turn to mold and release back to the earth. This is going further back, just to show some other work that I've done around ritual, and trying to weave parts of things I've carried with me, stones that I'm wearing, to connect to, this is a story about my grandma, but I'm using this found form. And then there's like one last slide of just the portal that opened it all. How come home, I'm home. This is what brought me here, and this is the origin of Mother. Thank you. Like what do the teachings mean kind of being passed? Is that what you said?
I guess I'm just curious like what it means to be regenerated.
I think I'm still trying to figure that out. I don't know yet. I think like in that, like when I first like made a form of myself, that was like the plaster cast mold. I was like, oh, this looks like me. And people are like, oh yeah, that is you. But I'm like, but it doesn't feel like me. So I guess there was like the severing of self, but then starting to like collect the pieces and like layer them at SCOBY made me feel more connected to it. And I guess it was just like, oh, this is kind of fleshy, this feels like skin. And now I'm like layering this like plaster that is like back to the earth and like it won't ever break down, but I'm layering something that will break down. So I guess it was this kind of like layering and unlayering, knowing that parts can like decompose, while still trying to like preserve this artifact. That's something that like has come up for me.
Do you think that like our close relation to beasts genetically made the process a bit more familiar with working with SCOBY? Because you talk about how plastic is bad for the earth and how it has very different materiality, but SCOBY resembles so much of human form, right? Both like how it's composed and how it feels.
I think so. Like calling like these microbes forward, I'm like they're already like inside of me because I'm consuming them. They live in my like gut microbiome. So it's like the memory to me is like they are still there. And I've been questioning like, are they kind of instructing me to like do these things? Are there like messages that like I don't know how to like directly communicate, but like are they still passing messages? That's something I've been like trying to like move forward with research of like the connection between like the gut and the brain, and like how does that influence like how we even sense like what the world is and like what the world is to us. So I definitely think like there's a connection between that of like if we are consuming this organism, it is part of us. It is like changing our thoughts in some way, just like at a chemical level. But I'm not there yet. But
I
yeah.
have a follow-up.
Yeah.
So what's the process then with healing the word also as opposed to from
Was it what? Healing? I think
Talk
so.
about,
Yeah. Like I think like showing this work has been like even today I'm like, is this enough? So that's still like that trauma of like, am I showing enough? Is this like too vulnerable? Is this something that people can connect to? So I think like through the healing that like the layering and unlayering has been like this is something I'm carrying, this is something I'm releasing. And if someone resonates with it, that's cool. If people don't, that's also cool. But like we can touch some hearts and like yeah, I think like the part that you're asking about, a lot of it has been about touch and like trying to reconnect to my own body through touching something that resembles myself. I think that part feels healing. You first.
You did briefly show a slide about how wolves grow and how it resembles community. Has it resembled community from your point of view in any way?
That's the way the mold is growing. I would say yes, because even as Alvaro said, like the cross-contamination, like, I was like, oh, there's bioplastics growing over here. If I'm working over here on this bench, like, I'll be fine. But, like, coming back to the work and being like, oh, it's completely, like, consumed. And I think that was a question that I was like, oh, what do I do with this? And someone in the lab, like, next to I was like, was the mold welcome? And that made me think about, am I welcoming in this thing to this community to kind of think about how do I fit into community? How does this fit into community? And like, yeah, I think I'm still figuring that out.
Yeah, because like to me it resembled colonization, which you mentioned previously as well. So I was just curious, like since it's your work, like how does it feel from your perspective?
That's a good point. Like I think that there's parts where like even coming into the lab, I'm like, I don't recognize these forms. There was a severing. Like I've grown these scoby, but I don't know them. And like even thinking of like biotechnology, it's like are we still using like colonizer tools to reshape the future? Yes. But like how do we think past that? So I think it's connected. Like we are all connected to like colonization. I'm still trying to figure out like where the like parts woven tightly and like where branches kind of like thinner that I can like examine the parts. Like a lot of this has been like nestled like bunches of nerves tied so tightly that I'm still trying to like unbind them. So it's like even looking at like the SCOBY growing and like the mold colonizing it, I still can't separate like the beginning and the end. Is this colonization? Am I also part of like adding to that form? Does this have agency? Or I'm kind of like instructing it to do this thing? Yeah. Yeah.
So I noticed a protocol and I'm wondering if it was intentional or not. When we're talking about like layering the story with others and like the mitochondrial DNA that would not like change the natural DNA of one both. Is that like an intentional influence or is that just something?
But isn't that that's like underneath it? When I was in school I started to learn about it. But then when we were doing the workshops, I was like, oh, like there is this thing. We did this workshop here where we started to learn about our mitochondrial DNA. And it's still something that I haven't like digested because science is just big parts. So I'm just trying to take small bites of it, but it's there. So it's like the things you're picking up on, yeah, it's there.
So one more question.
So I really like your ritualistic approach to your project. And ritual isn't a term that you often hear in modern Western science, or at this point you could say modern science. So how do you think modern science could benefit from the same approach?
That's a good question. I think like, I don't know, even being in this lab, there's this like being very sterile. It's like who is this sterile for? Who is this like I don't know. I think there's a lot of like just words that science uses that you're like, oh, but could we be softer with that word? Does it have to like use words like, oh, this thing is like going to like to be like crass, penetrate this thing to make this form happen? But it's like, how can we use different words? But I think like it's making me aware of like the vocabulary that we're using to kind of like shape this language. Of like vocab shapes how we connect to something. And if science is kind of like this distancing from, I'm asking like how can we be close to? How can we touch more? But like put our own vocabulary into that. So I guess I'm still learning like if I'm weaving in ritual, where do I start learning my own words? And like where there's like words of other people. I'm still like trying to find that balance. Thank you.
Thank you again. There's this beautiful weaving of these two artists and coming together in sort of their themes and their practices. So we're going to take a little break. People can get more snacks or go to the bathroom. I know I have this light right in my face.